Hello everyone, meet my dragon Clementine. He's the inspiration behind my single dominant epistasis video today. Before we get into single dominant epistasis, there are a few terms I would like to go over. First, a chromosome is a thread-like DNA molecule that carries genetic information. Alleles are variants of genes that may impact the phenotype of an organism. A locus, or loci for plural, is a location on a chromosome. Phenotypes are the physical characteristics, for example, the color of an organism. Genotypes are genetic characteristics in which there can be dominant or recessive genotypes. Dominant ones are shown through capital letters like big A, or big A heterozygous. Recessive genotypes are shown through lowercase letters such as little a. Genotypes can be homozygous or heterozygous. Homozygous has two identical alleles on genes such as big A, or little a. Heterozygous ones have two different alleles on genes such as big A, little a. Next, I would like to go over what epistasis is. It is when the expression of alleles at one locus masks the expression of other alleles at another locus. Multiple different genotypes can lead to the same phenotype, and it is the interaction between two different loci. Single dominant epistasis is where the dominant allele at one locus masks the expression of alleles at a second locus. In this case, A is epistatic to B, meaning that big A something results in a certain phenotype no matter what is at the B locus. In this scenario, we will be focusing on the color of bearded dragons. We can go from a blue dragon to a yellow intermediate or an orange dragon. For this dihybrid heterozygous cross, we are going to be running through the genotypes I have listed on the board. For the first one I have circled in blue, we have big A something, so inhibitor A blocks enzyme 1, and we are going to be stuck at the blue dragon. The same thing goes for the next one I have circled in blue because it is big A something, so it will also be stuck at the blue dragon. We are now going to look at the next one I have circled in blue. Since it is little a, big B something, we are going to progress to the yellow dragon intermediate due to the fact that we have little a. And since we have a big B allele, we are able to progress even further to the orange dragon, the final product. Lastly, since we have little a, little b, we are only able to progress from the blue dragon to the yellow dragon intermediate because the little b blocks us from progressing any further to the orange dragon. This ends with a ratio of 12 to 3 to 1. Since this new cross is not a heterozygous dihybrid cross, we cannot depend on the 9 to 3 to 3 to 1 ratio. Once we cross them, we find that the following genotypes shown are possible. The first two genotypes that are circled in blue both have big A, thus inhibitor A will block the enzyme, causing both to be stuck at the blue bearded dragon. When we look at the next two circled in blue, we can see that they are both homozygous little a. This means that they can progress further to the yellow dragon intermediate. Now we can look at the B locus and see that they are both big B something. This means that they could both progress past the yellow intermediate and will be orange dragons. Our phenotypic ratio with this cross will be 2 to 2 rather than 12 to 3 to 1 like the other heterozygous dihybrid cross.